So to understand the hierarchy of a protein, we need to start at the basic unit. We need to start at the monomer. So when we think about nucleic acids, our base monomer is a nucleotide. When we think about proteins, our base monomer is an amino acid. So what exactly is an amino acid? What do they look like? How many are there? What's going to be happening with these? So what's going to happen is I would suggest that you pause and either download the file for this or go to, actually, if you're at school, I've already got these printed for you. Can you go to the lab and make sure that you've picked up your little pack of cards? Yeah, so there should be a pack of about 20 little cut up cards for you. And you'll need 2 colored highlighters to do this activity. I want you to spread out all the amino acids on your table, count how many there are. Can you group them in any way? And then I want you to color 2 different things. I want you to color whatever they have that's the same in one color and whatever they have that is different in another color. Now, it doesn't have to be colored all the way in. You can just do loose circles. Doesn't matter what it is. But if you haven't done that yet, please go pause and do this activity. Spend about 20 minutes to half an hour, making sure, maybe even less, I don't know, see how you go, and make sure that you can identify each one of these amino acids within, and see what patterns that you can find. It's a much more powerful tool than me just telling you how many there are and what's the same and what's different. So hopefully you paused, you went away and you looked, you did this activity, because now I'm going to tell you a bit about amino acids. So all proteins are made up of amino acids. I want you to think of amino acids like Lego bricks. So there are lots of different types of Lego bricks. You can have different shapes of Lego bricks, different colors of Lego bricks, but they all have things that are in common. They all have spikes on one side, like the little bumps, and they all have a hole on the bottom. Yeah, so amino acids are the same. They share similar features, but they can be a little bit different. And depending on what order you put them together, depends on what protein you build. So Lego is a really useful analogy to think about how we build proteins. So all proteins are made up of amino acids. Most of the amino acids end in an -ine or an -ine suffix. So methionine, serine, thiamine, sorry. And now I can't think of any of the others off the top of my head, yeah? Glutamine, yeah? So a lot of them end in that. There are a few that end in 8 as well, but it's a good indication. If you see -ine at the end, you're probably looking at an amino acid. Amino acids are the subunits or monomers that join together to make that polypeptide chain. So thinking back to translation and building the polypeptide chain using the amino acids on the back of the tRNA molecules, that's what we're talking about. I drew them as little circles in protein synthesis. Now we're going to zoom in on that circle and see what it might actually look like up close. Polypeptide chains are just that. They're just a chain. That's useful, but it doesn't do all the different functions that we need proteins to do. So they fold and they coil and they form different bonds, and that enables them to form different shapes. And that's what this lesson is mostly all about. So here is a basic amino acid. You can also reduce this COOH and this NH2 over here to just be this. Yeah, so you don't have to draw it as this. You don't have to draw this as this. So when I redraw that, you could draw an amino acid as an R with a C, COOH on one end and NH2 on the other end. Yeah, so you can draw it really, really simply. You don't have to draw all of these kind of bonds unless you want to. If you understand that, if you're a chemist, go for it. Yeah, this is a carboxyl group on the right, and this is an amine group on the left. So all amino acids have this same basic structure. These are just a few different ways of drawing it. As I said, it can be really, really simple. It can be a carbon atom in the middle. We've got an amine group, on one side, which is NH2. On the other side, we've got a carboxyl group, which is COOH, and up the top, we've got a variable R group. Now, if you remember back, carbon has 4 valence electrons. Sorry, this is a bit of a chemistry lesson. So it needs to form a 4th bond. So what will happen is there's a hydrogen that goes down the bottom there. All right, so this is the basic structure of an amino acid. Now, what we're going to do is normally I would pause the slides and I would get you to come up and try and circle some of these groups on here. Now, so I might get you to pause, have a look at this image and see if you can circle some amine groups or identify some amine groups just from these pictures. Can you identify some carboxyl groups? And can you identify any R groups? So I'll get you to pause and then we'll restart in a second. All right, so hopefully we're now restarting. I'm just going to focus on these amine groups to begin with, and I'm going to make them green. So you can see here's an amine group on this one. Oh, it's over here. Yeah, but every single one of these is going to have a good old NH2. The proline's a little bit different. That's a little bit of an exception, but you'd never have to identify proline. The other thing that's on each one of these, there should be a COOH. So there's a COOH here, here, getting there, all the way through all of these. Yeah, and the final thing that all of these have is a variable R group. So in this case, remember one hydrogen is meant to be there. So anything that's not the one hydrogen coming off the C. So this one's R group is actually really big. Yeah, it's this whole section here. This one's R group is this whole section here. This one's R group is everything. Let's see if I can draw properly. Up like this, that R group's there. Our central carbon's down the bottom for proline. So that's all R group. So when we just have a look at this, what can we see that is the same? And what can we see that is different? So you can see that every single one, except for this proline down the bottom here, but ignore that. It's a chemistry issue, not a biology issue. Every single one has the same small NH2 group and the same small COOH. It also has a hydrogen off its central carbon atom. Yeah, so every single one of them has those three features. What does change is that blue section. And that blue section is what we call our R group. And that is what determines what type of amino acid it is. So I'm hoping that when you went through your amino acids, you discovered that there were 20. There are 20 different amino acids. And that means there are 20 different configurations of R groups. Now, of course, there's infinite, but on Earth, all we know about are these 20 amino acids. And so here's just two more examples. The simplest one is called glycine. It looks like this. It's got a hydrogen as its R group. It's got a carboxyl, and it's got an amine group. Okay, so the R group in this one is simply one hydrogen atom. Yeah, this one is methionine. Now, if you remember, methionine is the start codon, which means it's in every single protein. So it's got a carboxyl group, it's got an amine group, it's got a hydrogen poking off the top there, and it's also got this bit here. Okay, this is called its R group. All right, this R group has a sulfur in it. Yeah. Now, if you were to get really deep into the chemistry, you could understand why it's got the sulfur, but essentially all we need to know is that they're different. Yeah, and that's all I want to leave you with, is that there are 20 different amino acids. They all have some similarities. Yeah, they all have some similarities, and they all have a few differences. And that's your takeaway from this part of the lesson.